Hello, welcome to World of Microbes. Today we are gonna learn about microbiology laboratories, techniques, key activities in microbiology laboratories, and good microbiological practice. A microbiology laboratory is a laboratory devoted to identify microorganisms including bacteria, fungi, yeasts, parasites, etc. The microbiology laboratory has a crucial role in effective infection prevention and control. This lab covers basic concepts of microbiology with an emphasis on sterile technique, examination, microscopy, culturing, isolating and identification of microorganisms. The isolation and characterization of microorganisms inflicting infections performed by the microbiology laboratory play two important functions. Number one is clinical, in which a microbiologist has to test specimens from patients for microorganisms that are, or may be, a cause of the illness and to provide information, when appropriate, about the in vitro activity of antimicrobial drugs against the microorganisms identified. Number two is epidemiological, in which laboratory diagnosis is an essential element of communicable disease surveillance, both for routine confirmation of infections and for the rapid identification of the cause of outbreaks and epidemics. Surveillance is the ongoing, systematic collection, analysis, and interpretation of health data. Good microbiology practice, or GMP, comprising of aseptic techniques and other good microbiological techniques or practices, are of utmost importance to prevent contamination of the laboratory when handling microorganisms. GMP employs microbiological lab techniques, or microbiology lab procedures to keep the research materials, microorganisms, in their safe place without coming in contact with other organisms, and also keeping the safety of the people around in mind while experimenting. The ultimate goal of GMP is to extract out the research result without any adulteration or contamination. Microbiological techniques. Microbiological techniques consist of an aseptic technique, that is a procedure that is performed under sterile conditions to prevent the introduction of unwanted organisms or bacterial contaminants into an environment. This is an important microbiological lab technique to avert any contamination of laboratory personnel, cultures, and equipment. There are three processes to perform aseptic procedure, that is sterilization, disinfection, and sanitization. Sterilization is the decontamination process, in which all forms of microorganisms and their spores, for example, bacteria, viruses, prions, are killed. This is usually carried through physical methods such as heat, radiation, and filtration, or by chemical methods. Disinfection is also a decontamination process that is used to eliminate or inhibit the growth of bacteria or other pathogens, microorganisms, from inanimate objects or surfaces. Disinfectants such as alcohol, chlorine, etc. are utilized to make the substance and surface infection-free. Whereas sanitization aims at killing and eradicating the bacteria, pathogenic, and non-pathogenic microorganisms that grow on a surface top. A large number of products and chemicals are employed for the sanitization purpose, namely chlorine-based cleaners, alcohol-based cleaners, formaldehyde, hydrogen peroxide. For culturing a microorganism, a small sample is introduced to a culture media, usually with the help of a platinum wire probe streaked across its surface. The growth that is observed on or in the medium is the culture. There are 5 basic microbiology lab procedures, 5 I's, that are utilized by the microbiologists to examine and characterize microbes, namely inoculation, incubation, isolation, inspection, and identification. The technique through which a particular microorganism is introduced into a culture medium to develop the colonies is known as inoculation. The most common method used for spreading is either by a heat-sterilized loop or a micropipette with sterilized tips. The pour plate method, spread plate method, and streak plate method are all included in the techniques. Once the inoculation is done, the agar or the broth containing the inoculum is kept in the incubator at a certain temperature for the required time duration. This supports the optimum growth of bacteria. In microbiology, the term isolation refers to the separation of a strain from a natural, mixed population of living microbes, as present in the environment, for example in water or soil, or from living beings with skin flora, oral flora, or gut flora, in order to identify the microbes of interest. The inspection and isolation takes place through various methods like morphology, staining, implemented to identify microbes through their cell properties, using microscope. Some microorganisms such as viruses require an electron microscope to be seen, and not a simple microscope, biochemical tests, and molecular analysis.